Look, if you're a 15-year-old biology student gearing up for that massive test, you are in exactly the right place. Today, we're building the ultimate cheat sheet on biomolecules. I'm talking about the literal building blocks of life. My goal here is to make absolutely sure you walk away with a crystal clear understanding of the chemical foundations that make you, well, you. We're going to tackle everything from the chemicals in the food you eat to the actual genetic code inside your cells, so you are 100% prepared to ace this exam. Let's dive in.
We've got a solid roadmap today. We'll be hitting 5 key areas: the tissue analysis, primary and secondary metabolites, carbohydrates and lipids, proteins and nucleic acids, and finally, enzymes.
Section 1. The tissue analysis.
So to kick things off, we have to ask a really foundational question. How do we actually analyze living tissue? If you see this pop up on the test, you need to know exactly how scientists physically separate the components of, say, a plant leaf or a piece of liver in a lab to figure out its exact chemical makeup.
It actually comes down to a super simple 3-step grinding and straining process that separates everything into 2 distinct pools. Scientists basically take the living tissue and grind it up with a chemical called trichloroacetic acid, using a mortar and pestle. It creates this thick, wet slurry. Then they just strain that slurry right through some cheesecloth or fine cotton.
The best way to visualize this is to think of a basic coffee filter. The liquid that drips all the way through has all the tiny micromolecules. We call this the acid-soluble pool, and it's full of micromolecules weighing under 1,000 Daltons, stuff like simple sugars and amino acids. Now, the wet, chunky grounds left behind in the filter, that represents the massive macromolecules. This is the acid-insoluble pool, containing giant molecules weighing over 10,000 Daltons, like large proteins and nucleic acids. Now listen up, because here is a classic trick question your teacher might throw at you. Lipids are actually tiny micromolecules, so why on earth do they get stuck in the insoluble pool? Well, it's because when we grind up the tissue, the lipid cell membranes break apart and form these little vesicles that are not water-soluble, so they end up getting trapped in our metaphorical coffee filter.
Moving on to Section 2. Metabolites, primary versus secondary.
You absolutely need to know the difference between what a cell strictly needs to live and what it produces for other functions. This is incredibly high-yield exam material. Primary metabolites, those are your absolute non-negotiables. They are essential for the growth and reproduction of an organism, and you'll find them in literally every living thing. Think amino acids for building muscle or sugars for energy. Secondary metabolites, on the other hand, are not essential for basic growth. We mostly see them in plants, fungi, and microbes. A plant might use them for ecological defense, but honestly, their specific physiological roles aren't always fully understood by scientists.
But here's why that distinction is a total game changer for human health. Those secondary plant defense chemicals are actually isolated by scientists to make real-world medical drugs. We're talking about morphine for severe pain relief, vinblastine for treating diseases, and antibiotics to fight off bacterial infections. These so-called non-essential chemicals literally revolutionized modern medicine.
All right, Section 3, carbohydrates and lipids.
When you're studying carbohydrates, which, by the way, are literally just hydrates of carbon, you really have to understand their chemical skeleton. With only one exception, which is dihydroxyacetone, all monosaccharides contain a very specific carbon structure called a chiral carbon. It's basically a carbon atom that is attached to 4 entirely different groups of atoms, which makes it structurally asymmetrical.
Now, when those simple sugars link together into massive chains, they form what we call polysaccharides. And here's the key difference you've got to memorize for carbohydrates. They generally do one of 2 things. They either store energy, like the starch you'd find in a potato or the glycogen in an animal liver, or they build super tough structures, like cellulose in plant cell walls and chitin in insect exoskeletons.
Okay, let's pivot to lipids, or fats. Pay really close attention here. Saturated animal fats metabolize into cholesterol, which can actually block blood vessels, acting as a major diagnostic warning sign for cardiovascular disease. Why? Well, saturated fats have only single bonds between their carbons. This makes them straight, easily stackable, and totally solid at room temperature. Think of a stick of butter. Unsaturated fats, however, have double bonds. That double bond puts a literal kink in their chain, making them liquid at room temperature, like olive oil. So limiting those solid, saturated fats is a huge part of maintaining long-term health.
Let's move into Section 4, proteins and nucleic acids.
So proteins are heteropolymers made up of amino acids, and amino acids are fascinating because they have both an acidic group and an alkaline group. This means they actually change their ionic charge depending on the pH of whatever environment they're in. At a very specific pH level, the amino acid becomes what's called a zwitterion. This means the molecule acts as a dipolar ion balancing equal positive and negative charges at exactly the same time.
Pretty cool, right? So how do these amino acids actually link together to form stuff like collagen, which is the most abundant protein in the animal world? They connect end-to-end using a peptide bond. This bond forms through a process called dehydration synthesis. That simply means that as two amino acids link together, a molecule of water is essentially squeezed out and released.
And you have to remember that proteins have a strict structural hierarchy. A protein doesn't just stay as a primary, boring, straight chain. It coils up into a secondary helix, and then it folds into a really complex 3D tertiary structure. Sometimes, multiple folded chains even lock together to form a quaternary structure, just like the hemoglobin that carries oxygen around in your red blood cells right now. And here's the kicker. If a protein loses that specific 3D folded shape because of extreme heat or wild changes in pH, a process we call denaturation, it completely stops working.
Next up, nucleic acids. You will definitely see this comparison on your test, I promise you. These two nucleic acids dictate our entire genetic blueprint. DNA is the famous double-stranded helix, built with deoxyribose sugar and the nitrogenous base thymine. RNA, on the other hand, is the single-stranded messenger. It uses ribose sugar instead, and it swaps out that thymine for uracil.
Finally, Section 5, enzymes, the catalysts.
Before any chemical reaction can yield a product, it has to pass through this unstable, really high-energy transition state. Enzymes act as biological catalysts by actively lowering this activation energy barrier. They make it so much easier for a substrate to turn into a product. Seriously, without enzymes, the chemical reactions keeping you alive at this very second would happen way too slowly to actually sustain life.
Think of an enzyme's active site as a perfectly shaped needle pocket designed for one specific substrate. It's a simple four-step cycle. First, the substrate binds to that pocket. Then, the enzyme basically hugs it tight to alter its chemical bonds. Third, the new product is formed. And fourth, it's released, meaning the enzyme is totally free to immediately grab another substrate and do the whole thing over again.
But knowing how to shut an enzyme off is just as important. For example, sulfa drugs actually use competitive inhibition to stop bacterial pathogens from multiplying. Competitive inhibitors are basically structural copycats. They look exactly like the real substrate, so they can jam themselves into the active site and block it directly. Non-competitive inhibitors are a lot sneakier. They bind to a totally different spot on the enzyme called the allosteric site. When they do that, they cause the entire enzyme to change its shape, which means the real substrate can't fit into the active site anymore.
So I want to leave you with a bit of a provocative thought. How do these invisible, microscopic chemicals completely dictate our human health, our survival, and our everyday experience? Understanding biomolecules isn't just about acing a biology exam, but you're definitely going to do that now. It's really about unlocking the literal chemical architecture of your own life. From the primary metabolites constructing your tissues to the complex folding of the enzymes driving your metabolism, every single breath you take is governed by these extraordinary molecules.
We're exploring the absolutely fascinating, microscopic stuff that makes up our bodies and the entire living world. We're going to strip away all the macro-level complexity of biology and get right down to the chemical foundations, the very molecules that build us, fuel us, and keep us functioning every single second of the day. Here is our roadmap. We'll kick things off with the recipe of life, move through carbs and lipids, check out proteins, the heavy lifters, marvel at enzymes, nature's catalysts, and finally, wrap up with nucleic acids, the blueprint.
So, Section 1, the recipe of life. What are the raw materials that actually make up a living organism?
It's honestly mind-blowing when you think about it. We are literally made of the exact same elements as the inanimate rocks beneath our feet, just mixed up in a completely different way. If you look at the Earth's crust, it's absolutely loaded with oxygen and silicon, but it has barely any carbon or nitrogen. But us, the human body is completely carbon-based. Our relative abundance of carbon and hydrogen is way, way higher. We are essentially taking the same planetary ingredients, but we're throwing them into a uniquely organic recipe.
So how do we actually figure out what we're made of? Well, scientists use what is basically a biological blender. You take a piece of living tissue, like a leaf or some liver, and you grind it up in a harsh chemical called trichloroacetic acid. When you strain that thick slurry through something like cheesecloth, it magically separates into two distinct fractions. You get the acid-soluble pool, which is where all your tiny micromolecules hang out, and the acid-insoluble pool, which catches all the giant, chunky macromolecules.
Now, as our bodies process these raw chemical elements, we create what are known as metabolites,
A primary metabolite is absolutely crucial for your basic survival, growth, and reproduction. We're talking about your standard amino acids and sugars here. But nature is wildly creative, right? So we also produce secondary metabolites. These aren't strictly necessary to keep a single cell alive, but they serve highly specialized ecological roles. Think plant pigments, deadly toxins like ricin, or even compounds like natural rubber and morphine. Moving right along to Section 2, carbs and lipids. Carbohydrates are your classic biological fuel and building materials. You've got glucose, which is the single most abundant organic compound on Earth and the main engine fuel for our cells. Then you have the structural carbs, like cellulose, which builds plant cell walls and literally makes up the paper you write on. There's chitin, which forms fungal cell walls and gives beetles that distinct crunch when you accidentally step on them. And for us animals, we take our extra energy and pack it into glycogen, a highly branched molecule we safely stash away in our liver and muscles.
But then we hit lipids. And lipids, well, they are the ultimate biological rule breakers. Here is exactly why.
Remember that biological blender experiment we just talked about? By strict chemical definition, a macromolecule has to weigh over 1,000 Daltons. Lipids are actually incredibly small. They clock in well under 800 Daltons, so they should fall right into the micromolecule pool. But because they are hydrophobic, they absolutely hate water. When we disrupt a cell, those lipid membranes instantly clump together to form water-insoluble vesicles. This clever little chemical quirk physically traps them in the acid-insoluble fraction, effectively sneaking these tiny molecules right into the macromolecule club. All right, Section 3, proteins. These are the physical machinery and the heavy lifters of the cell. Proteins are heteropolymers, which is just a fancy way of saying they're complex chains built from a mixed alphabet of 20 different amino acids. Now, your body is an incredible manufacturing plant, but it does have limits. There are 10 non-essential amino acids that our bodies heroically manufacture from scratch. But the other 10, those are your essential amino acids. Your body simply cannot make them, which means you absolutely must get them from your food to stay alive. The real magic of a protein, though, is all in its shape. Its entire function is determined by how it climbs this structural ladder. It starts out as a primary structure, just a straight chain, like beads on a string. Then, hydrogen bonds pull it into secondary spirals or zigzag sheets. Next, it folds back on itself into a highly intricate 3D tertiary globular structure. And sometimes it goes a step further into a quaternary structure, where multiple folded chains lock together to form a supermachine, like the hemoglobin currently rushing oxygen through your bloodstream. And just to show you how massive of a role proteins play on this planet, get this. While collagen is the most abundant protein in the animal kingdom, holding our physical bodies together, a photosynthetic champion called rubisco takes the absolute crown for the entire biosphere. It's the enzyme responsible for yanking carbon right out of the air during photosynthesis. Which brings us perfectly to Section 4, enzymes, nature's catalysts. These are a very special class of proteins that make life happen at the speed of survival. To really get how enzymes work, you have to understand activation energy. Imagine a massive, unscalable wall completely blocking a chemical reaction from taking place. The substrates are just sitting there trying to get over that wall to become products. Enzymes are essentially microscopic bulldozers. They bind directly to the substrate and bulldoze that wall, drastically lowering the energy barrier so the chemical reaction can happen almost instantly. To put that bulldozer speed into perspective, let's talk about the number 10 million. Without the enzyme carbonic anhydrase, our bodies would naturally form about 200 molecules of carbonic acid an hour. That is way, way too slow to keep us breathing and alive. But with this enzyme present in our cells, that exact same reaction accelerates 10 million times, churning out 600,000 molecules every single second. No way we survive without that. But because enzymes are these perfectly folded origami structures, they're incredibly sensitive to their environment. Even a slight spike in temperature or a shift in pH can cause them to denature, basically unfold, which completely ruins their active site. Their speed is also capped. Once every active site is busy with a substrate, they literally cannot work any faster. Interestingly, we can actually control them using specific inhibitor chemicals that intentionally block their active sites. This is a brilliant trick we use in modern medicine, like giving someone sulfa drugs to competitively inhibit and starve out bacterial pathogens. And finally, we've arrived at Section 5, Nucleic Acids, the blueprint. These are the master instruction manuals containing the codes for every single protein we just talked about. Every nucleic acid is a polymer built from individual blocks called nucleotides, and every single nucleotide is a perfect 3-part puzzle. You got a nitrogenous base, a pentose sugar, and a phosphate group. These 3 parts link together over and over again to form the repeating, unbreakable backbones of our genetic code.
And the physical structures of these acids perfectly match the day jobs. DNA is our ultra-stable genetic vault. It's the famous double-stranded B helix, built with a deoxyribose sugar and the base thymine. It's designed for safe, long-term storage. RNA, on the other hand, is the nimble messenger. It's single-stranded, uses the standard ribose sugar, and swaps out thymine for uracil. Its whole job is to read the vault, leave the nucleus, and tell the cell's machinery exactly how to build those crucial proteins.
Which brings us to a really profound question. We looked at raw carbon, at tiny lipids clumping into membranes, perfectly folded protein engines, and strictly coded chemical blueprints. If our cells are literally just a highly orchestrated symphony of inanimate atoms and molecules, at what exact point in this microscopic architecture does the chemistry end and life truly begin? Something to think about.